let's look into the aggregated view of proteins in details. We'll start first with the home page, which shows the summary of the protein. Here you can see a brief view of the summary section, summary page. This essentially gives you the name of the protein, as in this case, spike glycoprotein, from which organism it is, UniProt accession, and all the other biological functions involved in this particular protein. Sometimes you can also see the EC number and disease associated with that particular protein, wherever it is known in UniProt database. The second main feature of this page is the 3D viewer MolStar, which shows you a representative PDB chain covering various segments of the protein. Here you can, the currently displayed PDB chain is highlighted blue over here, and you can see the details of the PDB chain. You can see the UniProt residue range, which it covers along with its coverage in percentage. For this particular PDB entry, you can go to PDBe entry page from this button. Also, to know more about the details, how to navigate and go to other different regions of the sequence and how we select our representative chain, you can find this information in the help text in the tooltip. In general, we use the PDB entry with highest number of observed residue from various regions of UniProt protein sequences. And you can navigate from these arrows by clicking on these arrows on the next region or the next segment of your protein. Here you can see the structure for the next protein. This is essentially an NMR structure, which you can see the different models, and you can click on these arrows to navigate the various models for this particular structure. Next, moving on, we also have a feature called Superimposition. Every week we run Superposition pipeline, and here each PDB chain is structurally aligned using gazamped software, and these structures are clustered together based on structural similarities. You can view these clusters for a given region from this particular by clicking this button. It will open a 3D viewer MolStar showing you the superposed structures for that region. You can also click on the button which is present below the structures icon. This again will open the 3D viewer of the MolStar showing all the superposed structures for any given region. To read more about the superposition pipeline, you can refer to this link. Moving on clicking on the button for superposition, you see the MolStar window. Here you see the different clusters. For example, for this particular spike protein, we have 7 different clusters for the segment 1, which ranges from residue 319 to 591. Each of these clusters correspond to a uniform structural conformation. These may or may not have biological relevance. By default, you only see the representative PDB chain. However, you can unhide the other chains by clicking this I button. You can also navigate into various segments by choosing this drop-down menu and selecting the corresponding region of your interest. All the data for superimposition is available over the link over here. You can find the rotation matrix and the clustering dendrograms at this particular link. We also have, while superimposition, we also align any bound ligand within the cluster. And this view you can find by clicking the button present below the ligand icon. This is called 3D view of superposed ligands. This opens a 3D viewer showing you the superposed ligands. Now this view is very helpful in highlighting all the common ligand binding site residues. On the right-hand side, you see the ligand and the various carbohydrates which are bound to your PDB chains. You can unhide any of the ligand which is not of your interest and change representation using these action buttons. Moving on. You also have these icons, which gives you a quick overview of the structural data available for your protein of interest. For example, total number of structures, total number of ligand bounds, how many macromolecular interactions are there, and similar proteins and various publications. You can download all this data by clicking on this download button. Present below each of these icons. And here you can download files like MMZip file, FASTA files, and even validation data. For many, in case of polyproteins, as the spike protein over here, you will also see towards the end a section called processed proteins. This essentially shows you a list of all the processed proteins which can be accessed, and they have their separate aggregated view, which can be queried using the pro identifiers from UniProt. For each of the spike protein, processed protein, you can click on this segment, green colored box, to view the structure in 3D in MolStar. And you have these icons showing you the quick glance of available structural data. For instance, number of PDB structures, number of PDB ligands, and number of interacting partners. And you can also view the dedicated processed protein page using this button over here. Between all these various sections, you can use the navigation bar and click on the corresponding tabs to navigate between various pages. Alternatively, you can click on each of these icons to view the relevant page.